Well, I guess when we go from talking about general Gen AI to talking about how it applies in the life sciences, we thin out the crowd a little bit. And I'm Matt McIlwain. I'm a partner at Madrona Venture Group, but I'm just absolutely delighted that you're going to get a chance to hear from one of the true leading thinkers and doers in life sciences meets data science. I got to know Geoff von Maltzahn 2.5 years ago. He came to Seattle, where we're based, for an amazing Intelligent Application Summit, and he's going to share for the next 12, 15 minutes some overview of what's going on in the space, and I'm going to get up and ask him a couple of tough questions. So without any further ado, Geoff von Maltzahn.
So it's wonderful to be here. I'm going to talk a little bit about generative AI meeting the world of biology. I'm going to selfishly start with some of the stuff that I'm working on, but Matt and I are going to jam on some of the reasons why these two streams of fluid are well suited to one another. So this is self-evident. Everything around us is either a gift of Mother Nature or it's a product of human ingenuity, extracting principles, creating tools, and building stuff. And while that's made amazing progress, the progress in the life sciences has been, relatively speaking, far less amazing, in that virtually no biology works in ways that our brains are well suited to understand. But fortunately, much of the future of biology is not going to have to rely upon just what we can conceive of as principles within our minds. That future is going to be thoroughly generated. And the implications for medicine, for our food system, and for the sciences at large are amazing. And I hope to give you a little glimpse of that. So a reason for that is that there isn't a life scientist on the planet that could glance at this slide and quickly tell you whether any of these paragraphs of DNA would cure you, kill you, or win you the Nobel Prize. And what paragraphs of DNA often do in nature is they transcribe a sequence we call an RNA or an mRNA. That then dictates the sequence of amino acids that then folds itself into a structure that we call a protein. And that protein serves one or more quantitative functions, either in biology or in the world of medicine. And while everybody knows that DNA produces proteins, the longer you've been a biologist, the easier it is to lose track of how profound it is that the left side of this slide and the right side of this slide are connected. What that means is that in every single one of hundreds of billions of dollars of annual antibody therapeutic sales, peptide, protein therapeutic sales, all of the quantitative properties of those products are encoded from the sequence of DNA from which they arise. And if we could read that code and actually understand what those properties and functions are, then biotech would change forever. So in the little animation that you saw here, we wouldn't go fishing for things that can stick somewhere on a target, but you'd be able to ask, what is the DNA sequence of a protein that would bind at this spot, at this spot, at this spot, or this one? So 6 years ago, we started a company called Generative Biomedicines that we're partnered with March Capital on. And we had, at the time, what seemed like a wacky idea that maybe we should set aside physics and apply machine learning approaches to extract the principles that dictate some of the fundamental properties of the world around us. Our idea was that if you can generate uncountable numbers of human faces of people who have never existed, perhaps the same would be true at any length scale of biology. That without mastering any of the physical properties of a human hair or human eyeglass in the previous slide, machines have mastered what it means to be a human face with sufficient acuity to generate uncountable numbers of new examples. What if the same is true for every biomolecule that we use in medicine? What if the same is true for every subset of the biology that we enjoy on a daily basis? So particularly, we started in regimes that are rare in biology, where we had two large, high-quality data sets with which to direct machines. One of those was the hypothesis that is akin to the modern human large language models, that maybe if we could let machines just read every sequence of DNA we could get our hands on that encodes a protein, it might gain proficiency or fluency or mastery of what it means in the language of DNA to encode proteins with various properties. The second was using protein crystallography data sets, where over the past several decades, nearly every other week, the cover of Science and Nature has been the display of another Herculean feat of solving a crystal structure of some important protein in the world around us. And we imagined that maybe we could learn from those the principles that dictate the most valuable thing in all of biotechnology, which simplistically would be to be able to generate a protein to stick to a target in a desirable location without sticking to other stuff. That is what all antibodies do, all peptides do, and it's what most proteins do in medicine today. So we hypothesized that maybe the way that amino acids like to interact with one another inside of proteins could give us a glimpse of the way that they would like to interact with one another on the outside of a protein, and thereby possibly open up an ability to start generating biomolecules in those categories. So publicly available data sets gave us an enormous head start, but they weren't nearly the keys to the kingdom that mining trillions of approximately free tokens in human language on the internet has been for the large language models directed towards human languages. In fact, they sort of gave us the ability to start to crawl, to generate with low frequency binders that would bind with low affinity to a desired target. To get this to really work, we've had to build the equivalent of an embodied AI, or a biotech lab built so that the entirety of what we are testing are generated sequences coming out of the machine to various tasks that we're orienting it towards. We've now had millions of proteins flow through the circle that you see here, where a generated sequence is built, quantitatively tested, and every one of those successes and failures flows back into the models themselves, so that with each iteration of this cycle, we have the capacity to get wiser. And that's allowed us to do some things that to us have been extraordinary. I'm going to take you through a couple of those examples. So this is an example from a few years ago. We challenged the team to take the top $50 billion of antibody therapeutic sales and generate antibodies that would hit the exact same target, same epitope, same binding pose, same structural conformation at the interface, with comparable or higher affinity without going anywhere near the parent intellectual property of those molecules. Within 3 months, the team was able to do that for 100% of them. To put that in context, if you had all of any major biopharmaceuticals company's capabilities today, you wouldn't be able to do that in 10 years with prior approaches. You would find things that stick to many places or other places. But what I described is like Robin Hood splitting the first arrow with an arrow that goes straight through it 50 times in a row. What that means is that every time a target or a spot on a target shows a benefit to patients, there's the potential to create the very best version of a therapeutic for those patients. Oftentimes, a pioneering antibody or a pioneering therapeutic has liabilities. It either has manufacturing liabilities or limitations that require it to be dosed more frequently than would be desired. So imagine if every time we could direct the perfect or an ideal medicine to patients. We've also been able to direct antibodies towards things that have been difficult for immune systems and other systems to find solutions for. This is everybody's favorite acronym, COVID. And the reason we keep getting sick with COVID is there are portions of the spike protein that have a simple function, which is to be like Velcro to the ACE2 receptors. And they keep changing. So our prior infection doesn't help us very much with the wildest variants that keep pouring over us. Now, it happens that there are other portions of the spike protein that haven't changed almost at all, from COVID 1.0 to the wildest variants we've seen today. In fact, you can march up the tree of life in viruses to other SARS families, and these domains are still in common. But our immune systems are terrible at finding antibodies that can neutralize those domains that haven't been varying. Fortunately, generative AI has been helping us here. So we've now dosed healthy volunteers with the first generative AI invented antibodies that are able to hit those domains. And what that does is the portion of the spike protein that we're hitting is this super interesting machine that forces two membranes together so as to put the virus's genetic code inside of our cells. If you can bind to that domain and prevent it from acting as a mechanical vice to force those membranes together, then you can prevent the virus from entering into our cells. And our hope is that this is a glimpse of the potential for generative AI to provide solutions to some of the worst infections that we still face in medicine. We've also been able to start sort of bringing functions from one protein into another one in ways that, if you're familiar with how hard it has been to do anything in biology, to us are breathtaking. So this is one example. This is a microbial-derived protein called uricase. If you have gout, a microbial-derived uricase could be beneficial in that it can bind to uric acid and metabolize it. What you're seeing there is uric acid precisely localized in the enzyme active site. And enzymes are these amazing machines that grab onto a molecule with often sub-nanometer precision in every aspect of orientation and are able to catalyze a favorable downhill energetic reaction. The problem is, if you're a patient and you're administered a microbial-derived protein on a regular basis, your immune system does not like that. And many of these patients have severe adverse responses to them. So in partnership with Amgen, we've been asking, can we take the active site of uricase and plop it into the middle of a human protein like albumin, one of the most abundant proteins in our serum? Now, of course, it doesn't fit, so we've needed to apply our generative models to reconfigure the parent protein so that, in fact, you can establish angstrom precision with which to be able to localize that substrate. And this is one solution, but there's a whole bunch of solutions. Back to the antibody example, where something that was very scarce, we're now finding an abundant set of solutions for by being able to make predictions in function space and map them back into the sequence with which the protein can be synthesized. This allows you to then optimize for other parameters that are important for therapeutics. So I gave you an example of how we're doing applying this to new antibodies. I gave another example of how we're applying this to new enzymes. The same models that allow us to be able to do those are also applicable to the design of peptides. Many of you may be aware of the GLP-1 phenomenon of Mounjaro and other now household name therapeutics helping people to be able to lose weight. Generative models might be able to offer some value there as well. I've also applied these to proteins on the surface of cell therapies to help them be able to go after cancer more efficiently. We've been able to create multi-protein complexes with the potential to make things like gene therapies safer and more effective. And I want to leave you with the idea that has been swimming around in our imagination for several years. So we started the company 6 years ago, and this seemed like it was a worthwhile pursuit, but we had no real evidence that this was going to work. We're now convinced that 100% of protein therapeutics are going to be created this way. And the rationale is sort of simple. In any of the protein categories that I described, you have 60 to 100 amino acids that are playing a meaningful role in that protein's structure and function. Every one of those positions, you have 20 options. So that is 20 to the 60 to 100 or even 1,000 plus power, depending on how large a protein is. That is atoms of the universe to some exponent. And as you can imagine, biotech labs today and all of Mother Nature's ingenuity haven't had all the atoms of the universe at their disposal with which to be able to experiment. And oftentimes it's tempting when there's a new technology to imagine that it might just tweak what's possible. But I think that's the wrong mental model here. In that even if you make very generous assumptions around everything that Mother Nature has ever had a chance to test, of course, the vast majority of that died off and was not viable. All of those combinations represent less than one drop of water contrasted with what is combinatorially possible, which is as vast as all of the oceans on Earth. So just think about that for one moment. I think it's beautiful. Like everyone we've ever loved, everyone in this room, every life form that we've ever enjoyed, this majestic tree that we're near, everything that has come before us is in that one drop. tools like these are going to allow us to be able to navigate the seas, and we have no idea what's going to be out there. But it's fair to imagine that we're just at the very, very start of what biology can do. Thank you.
Great job. Well, it's a little daunting to follow that overview, but let's take this concept of the drop of water and all the other water, and there's probably two categories of tools that you're going to need, and I'm using tools very broadly. One is additional data, and then all these different AI tools. So let's start with the data. What data is still missing? What are the big categories of data that are needed so you can go from this virtually infinite set of possibilities that can be computationally, explored to get to things that are more on target?
Yeah, so I described on one of the slides that we started with a rarity in biology, which is to have two data sets of very high quality and which are massive. Those two data sets are all genomes that have been sequenced, where the field's obsession over high fidelity sequencing and low cost sequencing has given an extraordinary trove of information about the languages of life. The second one is three-dimensional crystal structures, where angstrom level precision has been a necessary component of the cover of Nature and other laudable demonstrations. The vast majority of science and life science are not like that. And so the advantage that the human large language models have had with extraordinary quantities of cohesive data with which to be able to utilize is almost entirely absent in the sciences. Oftentimes there are legal restrictions that make medical data sets and other data sets inaccessible. There are, even in published data sets, large quantities of data that are missing, that are necessary to make any sense out of what that data really means. Formats of data have changed substantially. So there are a whole eras of data that are sort of sitting on shelves, underutilized and unutilized by these sorts of systems, and it's going to take a lot of work to get them to be able to be unified in any cohesive fashion. Furthermore, depending on what your aspiration is, reading every paper that humans have written about science to date is not the way to master science. That's a way to extract and efficiently be able to interact with humanity's knowledge to date, but most of what we've been writing about in papers is a tiny glimpse of how biology really works. A big chunk of it is wrong.
Yep.
And therefore, part of the fun of any AI meets science endeavor is that you have to pay just as much attention to giving the AI access to the real world, allowing it to be able to run experiments, and all of that is really expensive, but it allows you to then blaze a trail where large portions of the data that allow you to achieve your ultimate technological feats are entirely proprietary to the companies that are doing it.
And so that sets up kind of an iterative loop. You showed your loop of, to oversimplify it, you take some data, you build some computational models, you explore a lot of space, and then you actually do things in a wet lab, probably a highly automated, increasingly digitized wet lab that then feeds back in to the loop to refine your models to try, to make the predictions and try the next things. What, if that's directionally correct, what are you using from an AI perspective to help you in the loops that Generate Bio, other companies that you and Flagship are involved with? What are you finding more useful? Can you take models off the shelves, or do you really have to build your own models? How do you take advantage of something maybe like an AlphaFold versus, some domain-specific models that you're finding you need to?
Yeah. So there's two categories of models to vastly oversimplify that I think are going to be incredibly important and synergistic in life sciences. One is the transformer architectures, human large language models, and roughly the same sort of force multiplier that many talks today have talked about them being directed towards the world of legal contracts or the world of enterprise solutions are going to superpower the life of every scientist on Earth, and allow things that have formerly been really cumbersome in the lab, like hiring a team to be able to implement an automated process, which usually is brittle, cumbersome, takes a long time to program the robot, and its ability to navigate any variation in the protocol that you're inserting to it is zero. That changes if a human large language model can go from human intention to experimental design to writing the software to program the robot with which to be able to conduct the experiment, and depending on whether the LLM has signing authority, at 3 a.m. order $100,000 of new reagents to do it again. The second category are models to actually understand how life works, or to create better technologies. What I described, we're relying heavily on diffusion models with which to be able to depict what an individual protein and what protein complexes will do. And the combination of those two is going to be increasingly powerful. You know, in that many times if one creates the ability for the model to design the next experiment, both in a sort of while loop to achieve some therapeutic feat, but also to improve the model, to take areas of high uncertainty, run a bunch of experiments to try to diminish uncertainty in the model, science is going to start to be conducted autonomously with some of these powers.
And how much of that do you effectively have to do yourself, or are there things, you know, I referenced AlphaFold earlier, for example, which is a protein folding model. Are you finding valuable to incorporate, so not just the outside data, but some of the outside models?
So AlphaFold's a great example. It's a machine-learned rules of protein folding that came out of DeepMind several years ago. And it's fair to say that it has been a revolutionary tool in life sciences, in that all of that painstaking protein crystallography work I described is bypassed for millions of proteins. There are still edge cases where it struggles. But I think that one needs to be voraciously interested in using off-the-shelf models, so you can play around and figure out what the strengths, weaknesses, deficiencies are of various approaches. Fortunately, there's a large community of open source biomolecular modeling efforts akin to some of the open source efforts that are taking place on the human large language model side of things.
I'm curious, in the two different paths we can go down here. One is, what are you finding from a receptivity perspective amongst, we'll call it big pharma, and how are they leaning in? Are they skeptical? You mentioned Amgen working together with different people. Talk a little bit about that. And then I'm also interested in big tech. So big pharma, big tech, and how those two worlds are partnering together with you.
Yeah, so we have a relationship with Amgen at Generate. It's been incredibly fruitful. Amgen identified when we were a 30-person team that generative AI was going to be a lot more than just a 30-person startup somewhere in Cambridge, Massachusetts. And they spent time putting together what we surmised to be some of the hardest things that they've ever tried to work on, and threw them our way. And that's kind of like barbecue chicken for a team like us. It's created a beautiful relationship where there's a lot of idea flow back and forth. They have helped mature our organization a lot, and can help us in the process of going from a wild idea to a drug discovery organization and now a drug development organization. We have our first 2 programs dosing patients with antibodies that have come from our generative models. As you can imagine, there are plenty of skeptics as well. And I think that's just part of the game.
How would you kind of, maybe just to take another company that we're both, I'm sure, a little bit familiar with. So how do you think about like a isomorphic? This is a company that actually spun out of DeepMind and is working on different aspects of biology meets data science. Is there a different type of skepticism for a company that comes more out of the tech background than the life sciences background that also has a generative or capability set, an AI capability set?
Yeah, I don't know. I mean, I think every single profession is facing some version of, well, only I can do the following. And all of us are probably going to be closer to wrong than right with that inclination. Like we are going to have to intellectually migrate en masse to new frontiers and figure out, where a decade ago we put a filter in our head that is just wrong. I suspect they're facing some of the same opportunities and challenges where there's people with a twinkle in their eye that believe, the evidence in front of them, or others that believe that despite the amazing progress thus far, there's sure to be some brick wall just around the corner.
Yeah, one of my observations with them is they're less focused on trying to create that dry lab, wet lab loop inside the company. And I think that's a different approach than yours. And it'll be interesting to see if that, I tend to favor your approach, and we've got companies, Teray and others here, that have taken a similar mindset towards that and seen a good bit of success. Let's come back to the tech side of it. NVIDIA has been investing in life science companies. Google, of course, has got its arms, Amazon, even Microsoft. How have you found partnering with them? I mean, they've got the compute, they've got the massive scale. Have they been resources to you, or have you kind of gone and mostly done these kinds of things yourselves?
They've been incredible resources. And I don't want to depict them as only that.
Yep.
Because, it's very obvious to everybody here that NVIDIA and others enabling compute expansion is an absolutely necessary resource. Microsoft and others have provided the ability for models to reach the world with a speed that might not have been feasible otherwise. The open question is how many of these companies are going to start to wade into biology? Like Google has their DeepMind effort. DeepMind has done some amazing things. There has never been a better time for a tech company to strategically prioritize trying to master the rules of science and of life. But there's a lot of surprises too. And mental models like, we don't need to build a lab, we can learn everything from existing data sets. There are places where that's not viable in
On
life.
the generative side, I was with one of those companies for dinner last week. I think they're trying to figure out at that abstraction layer, could they be sort of the master agent, as it were, and there'll be lots of other models that serve up into kind of an agent architecture. We see this in lots of different domains. Does that seem pragmatic as an approach for them? Because they don't really necessarily know and understand the science quite as well as you and others do. Or do you see some other approach that you would sort of, if you were giving advice to Satya or Andy or Sundar, what would you say? Well, you should focus here.
I mean, given that I really enjoy the things that I do, I would try to pull them onto the mats and wrestle a little bit. I mean, I think if you buy what I concluded with, which is that we're at the very, very start of biology, and there's a variation in that calculation for material science, there's a variation in that calculation for chemistry, then there are going to be a lot of ways of creating value from an upcoming scientific revolution. I am most excited about the ones that produce solutions that cure diseases, prevent diseases from ever happening, or radically expand our access to materials that can do sensational things for sustainability, for the planet, for the world. And but to blaze trails like that, it requires
partners.
Many variations of partners and picks and shovels, and there's going to be a lot of ways to win.
I could ask questions all day of Jeff. Thank you all for being here. Thank you, Jeff, for your perspective and this vision of where we're going. Super excited to see it more and more as reality.